Good morning. Good morning to everyone. How are you all? I am also fine. Thank you. Okay. Right. Shall we start? So check once my audio and video. Please check once my audio and video and respond. Is it clear? Yeah. Yeah, everything is clear. Good. Okay. So let us recall the important points briefly before we are going to the electron transport system, right? What is the connecting link compound between glycolysis and Krebs cycle? Anybody? What is the connecting link compound between glycolysis and Krebs cycle? Very good. Acetyl coenzyme A, correct? Acetyl coenzyme A. How many carbons are present in the acetyl coenzyme A? How many carbons are present in acetyl CoA? Very good. 2 carbons. 2 carbons. Right. What is the place in the mitochondria where Krebs cycle takes place? What is the site of Krebs cycle? Yes, very good. Matrix. Matrix is the site of Krebs cycle in the mitochondria. Mitochondrial matrix. Okay. What is the first product of Krebs cycle? What is the first product of Krebs cycle? Yes, citric acid. Citric acid. Or simply you can say citrate. As citric acid is the first product of Krebs cycle is also known as citric acid cycle. Citric acid cycle. 6 carbon cycle. Because citric acid is a 6 carbon organic acid. Very good. Next. Is it Krebs cycle is cyclic pathway or non-cyclic pathway? Is it Krebs cycle is a cyclic pathway or non-cyclic pathway? Yes, it is a cyclic pathway. That is why it's called as Krebs cycle. Yes. To complete Krebs cycle, how many turns it must occur? to complete the Krebs cycle, how many turns it must occur? Very good, very good, very good. 2 turns. 2 turns. Okay. Then, in Krebs cycle, only one 5-carbon compound is formed. 5-carbon compound is the only one compound formed in Krebs cycle. Can you say what is the name of that compound? Can you say, very good, alpha ketoglutarate acid? Simply you can say alpha KGA. Alpha KGA. Alpha ketoglutarate acid, a 5-carbon intermediate compound of Krebs cycle. Right. In Krebs cycle, how many CO2 are liberated? In Krebs cycle, how many CO2 are liberated? Very good. 4 CO2 totally. 2 decarboxylations occur. Okay. For each decarboxylation step, 2 CO2, totally 4 CO2. Correct. Next. At how many places NADH are produced? NADH are produced. It is the reduced form of NAD plus. 3 places. Means totally, okay, 3 places. Totally, how many NADH are produced? 3 steps. At 3 steps, NADH are formed. At each step, 2 NADH. Totally, 6 NADH are formed. 6 NADH. I mean to say, each step 2, 3 steps, 3 into 2, 6 are produced totally. Very good. Okay. At how many places FADH2 are produced? At how many places FADH2 are produced? 1 place, 1 step. That means how many FADH2 are formed? How many FADH2 are formed? 2. 2. 2 FADH2 are formed. Very good. For more clarity, once again we are briefing the concept that we taught yesterday. Okay, that is the reason why I am recollecting all the points once again, right? Okay. So finally you can say 4 CO2 are formed, 6 NADH are formed, 2 FADH2 are formed. Okay. And it is a cyclic pathway. Finally you can say. What is the starting compound and ending compound of Krebs cycle? Anybody? What is the starting compound and what is the ending compound of Krebs cycle? Yes, OAA means oxaloacetic acid, or you can say oxaloacetate, oxaloacetic acid or oxaloacetate, a 4-carbon compound. That is the starting and ending compound. Okay, that's all. Right. Now we enter into the last step of aerobic respiration. Very important and somewhat a critical concept. So everybody should follow carefully. That is electron transport system and oxidative phosphorylation. Okay. Electron transport system, ETS and oxidative phosphorylation. Electron transport system and oxidative phosphorylation, in shortcut you can call it as OP, contrast to SLP. SLP means substrate level phosphorylation, OP means oxidative phosphorylation. Okay. Electron transport system. First of all, you have to know the meaning of electron transport system. Okay? Electrons are transported from one carrier to another carrier in a systematic manner called as electron transport system. So electrons are transported from one electron carrier to another, another to another, like this. As electrons are transported in a systematic manner from one electron carrier to another electron carrier, this is called electron transport system. Then what is meant by oxidative phosphorylation? Phosphorylation means formation of ATP from ADP and inorganic phosphate. Formation of ATP from ADP and inorganic phosphate is known as phosphorylation. If this phosphorylation takes place during oxidation, is known as, that means one compound undergo oxidation, then another becomes reduction. Now this compound undergo oxidation, then another become reduction. Because of oxidation reduction, energy is liberated. Using this energy, ADP combines with inorganic phosphate and form ATP. This formation of ATP is known as oxidative phosphorylation. So now you understand the meaning of what is ETS and what is OP. I hope you understand all. Yes. Clear? Now, where does this ETS takes place? And also OP. ETS occurs in the inner membrane of mitochondria. Inner membrane of mitochondria. This is the site of ETS. Very important point. I told you clearly. What is the site of glycolysis? Cytoplasm. What is the site of link reaction? Mitochondrial matrix. What is the site of Krebs cycle? Mitochondrial matrix. What is the site of the final step, that is ETS? Inner membrane of mitochondria. So this step takes place in inner membrane of mitochondria. For example, all of you know that this is the mitochondria. Try to recollect here. It is almost a sausage shaped structure. This is the outer membrane. Now see, this is the inner membrane. See, this is the mitochondria. Try to understand now. This is outer membrane. Outer membrane is smooth. This is inner membrane. Inner membrane shows infoldings. These are called infoldings. These infoldings are called cristae, plural. These inner membrane foldings are called as cristae. So on this inner membrane, ETS takes place. On this inner membrane, ETS takes place. This inner membrane contains several electron carriers like this. These carriers carry the electrons. First this gives to this one, this gives to this one, like that. That is why electrons are transported from one electron carrier to another electron carrier in a systematic manner. That is why it is called ETS. So this is the site of electron transport system. And all of you know that this fluid part, this one is called as matrix of mitochondria. Mitochondrion singular, mitochondria plural. Mitochondrion singular, mitochondria plural, right? Now see, this is the, so link reaction and Krebs cycle both takes place in the matrix of mitochondria, but ETS takes place on the inner membrane of mitochondria. Hope you understand. Right. Now let us see what happens during this ETS. Already we have seen that there are several reduced coenzymes are produced. Listen carefully. So far, at the end of Krebs cycle, you have seen many NADH and FADH2 are formed. These are called the reduced coenzymes. These are the hydrogen acceptors. NAD plus accept the hydrogen and becomes NADH. And these are energy compounds. They store a lot of energy. Understand? So, I told you that one glucose molecule produces 38 ATP in aerobic respiration. Correct? So far, in glycolysis, only net gain of ATP is 2. In Krebs cycle, only 2. These two are formed through SLP. So total ATP are 38. 38 minus 4, right? 4 ATP. That is equal to 34 ATP has to be produced. Got the idea? One glucose upon its complete oxidation produces 38 ATP. But actually so far, directly formed ATP through SLP, in glycolysis only 2, in Krebs cycle only 2, I am telling about the net gain. And total ATP formed directly through SLP reactions is 4 only. 38 minus 4, still 34 ATP has to be formed. Those more number of ATP, those 38 ATP are produced in ETS. So large number of ATP are produced in ETS. And one more point to remember, we are talking about aerobic respiration. Can you see so far the role of oxygen in any step? In glycolysis, it is independent of oxygen. And you didn't see the role of oxygen in link reaction. Have you seen any place else the usage of oxygen? No. In Krebs cycle also, no role of oxygen. But we are talking about aerobic respiration. We are talking about aerobic respiration, but still we didn't see the role of oxygen. No oxygen in glycolysis, no oxygen in link reaction, no oxygen in Krebs cycle. But you will see the role of oxygen in ETS. So, oxygen role is seen in the last phase of respiration, even though it is the crucial, it is the vital for continuation of the cycle. Understand the point? It is the vital step. So oxygen role is seen only in fourth step. And ultimate electron acceptor, the ultimate hydrogen acceptor is the oxygen, and the role of oxygen is found in electron transport system. That is the last step of aerobic respiration. And as I said, most number of ATP are produced in electron transport system. Now let us see how many NADH are produced so far. In glycolysis, 2 NADH are formed. In link reaction, 2 NADH are formed. In Krebs cycle, 6 NADH are formed. In Krebs cycle, 2 FADH2 are formed. Yes. Those are the energy compounds. Those are the energy compounds. These are formed after acceptance of the hydrogens. That means these are the reduced coenzymes. And these are energy compounds. when these are oxidized again, now these are in reduced state, when these are oxidized, energy is liberated. That means electrons are produced first. And again they become NAD plus. And these NAD plus again participates in Krebs cycle, link reaction. Understand? Again these are participate to continue the cycle. Remember, NAD plus becomes NADH in glycolysis, link reaction and Krebs cycle. In ETS, these NADH oxidized into NAD plus, FADH into FAD to continue the cycle non-stop, uninterruptedly. This has to be takes place. Students, you have to understand this is a very, very important concept. That is why I am telling repeatedly. It is not that easy. If you divert your mind, you can't understand comprehensively. So I will tell you once again, in glycolysis, in Krebs cycle, in link reaction, NADH and FADH are formed. That means NAD plus reduced into NADH, FAD reduced into FADH. Now, these NADH, FADH formed in these steps now undergo oxidation in the ETS. As a result, again it become NAD plus. So NAD plus become NADH in glycolysis link reaction Krebs cycle, and NADH become NAD plus. That means NADH become NAD plus in the ETS. So in order to continue the cycle uninterruptedly, all these 4 steps must occur continuous manner. Yes. Now we will see how these undergo oxidation and releases electrons, and these electrons are taken up by oxygen to form metabolic water. And in this step, how ATP are formed, we will see. Just I am telling the introduction to ETS. Finally, we conclude. What is the meaning of ETS? Electron transport system. What is the site of ETS? Inner membrane of mitochondria. What is there on the inner membrane of mitochondria? Several enzyme complexes are there. What they did? They carry the electrons from one complex to another complex. Who is the final acceptor of these electrons? Oxygen comes from external atmosphere into the body. That is the electron acceptor, hydrogen acceptor. Okay. Now most of the ATP are formed in ETS. What number of ATP are formed in ETS? 34. Remaining 4 are directly formed in glycolysis and Krebs cycle through SLP reaction. SLP means substrate level phosphorylation. Okay. Now we have to know what are the complexes present on the inner membrane of mitochondria. See, electron transport system. This is inner membrane of mitochondria. Inner membrane. Okay. Inner to the inner membrane, matrix is present. Inner to the inner membrane, matrix is present. Outer to the inner membrane, intermembrane space. Intermembrane space is present. This is called intermembrane space or perimitochondrial space. This is the inner membrane. So try to recollect. See, this is the inner membrane. Inner to the inner membrane, matrix. Outer to the inner membrane, intermembrane space. See, this is the intermembrane space. Intermembrane space, also called perimitochondrial space. Inner membrane, inner to the inner membrane matrix, outer to the inner membrane intermembrane space is present. Okay, I hope you understand. Now, on the inner membrane, 5 enzyme complexes are present. First one. See, this is the complex 1. I will tell you the names of the complexes also. That is the complex 1. Next, this is the complex 3. This is complex 3. Next, this is the complex 2. This is the complex 2. Next, this is the complex 4. This is the complex 4. And finally, the complex 5. Complex 5. This is complex 5. Okay. So once again, we'll go through this. Complex 1. Complex 3. Complex 2. Complex 4. Complex 5. This is complex 5. All of you see carefully, students. This is the inner membrane of mitochondria. On the inner membrane of mitochondria, there are several enzyme complexes are present. This is complex 1, complex 3, complex 2, complex 4, complex 5. 5 complexes. Now I will tell you the names of these complexes also. First complex. This is complex 1, as I said. This is the complex 1. This is NADH, dehydrogenase complex. NADH, dehydrogenase complex. And this is complex 3. Okay. This is called cytochrome BC1 complex. Cytochrome BC1 complex. And this is complex 2. This is complex 2. This is called succinate dehydrogenase. Succinate dehydrogenase. And this is complex 4. Complex 4. Cytochrome C oxidase. And this is complex 5. Complex 5 known as ATP synthase or F0 F1 complex. F0 F1 complex. This is F0. This is F1. Two components are there. This is called F0. This is F1. That is why it's called F0 F1 complex. Complex 1 NADH dehydrogenase. Complex 3 cytochrome BC1 complex. And complex 2 succinate dehydrogenase. Complex 4 cytochrome C oxidase. Complex 5 ATP synthase. These are the enzyme complexes. These are the enzyme complexes present on the inner membrane of mitochondria. Okay. Now, let us see what are the components present inside each complex. For example, in complex 1, FMN is present as a prosthetic group, flavin mononucleotide, and iron sulfur center is present. In complex 3, as I said, cytochrome B, cytochrome C1 and iron sulfur center. In complex 2, FAD is present, iron sulfur center. Yes, iron sulfur centers are present in 3 complexes. Complex 1, complex 2, complex 3. All of you following well? Are you following well or not? I am telling slowly in a detailed manner. What is the ultimate result of this process? To produce the maximum number of ATP. How ATP are produced? We will see. For that, I am explaining first of all the complexes. Okay, I told you 5 complexes, and now I am telling about the components of the complexes. These are enzyme protein complexes. And next in the complex 4, copper A, CUA, cytochrome A, cytochrome A3 and copper B. These components are there in the complex 4. Cytochrome A, copper A. I will write capital letter. Cytochrome A cytochrome A3, copper B. These are the components present in that complex. And the last complex in which electrons are not transported, only protons are transported, that complex is called ATP synthase. It is also called F0 F1 complex. F0 act as a transmembrane channel. F0 act as a transmembrane channel, that is a part of integral part of the membrane, and this is the head. This head is called the F1, and this is the catalytic site for ATP synthesis. Actually, ATP is synthesized in this part. This is the catalytic site for ATP synthesis. Hope you understand all. These are the 5 complexes. First complex is commonly called as gateway of electron transport. Gateway of electron transport is first complex. ATP is synthesized in the 5th complex. Only protons are transported, but not electrons in only one complex, that is 5th complex. In addition to these 5 complexes, 2 mobile carriers are present. 2 mobile carriers are present. One, ubiquinone. Second one, cytochrome C. Please students, please concentrate. Along with these 5 complexes, 2 more mobile carriers are required. What are those 2 mobile carriers? One is UQ, means ubiquinone. Ubiquinone. UQ means ubiquinone. And second one, cytochrome C. Don't confuse. Here I am used the word cytochrome C1. This is cytochrome C. Cytochrome C and cytochrome C1 are different. Okay. Now, the locations of these mobile carriers. Between complex 1 and 3, ubiquinone is present. Understand? And also between 2 and 3 are present within the membrane. Ubiquinone is present within the membrane between 1 and 3 and 2 and 3. That means it carries the electrons from complex 1 and transported to complex 3, and it also accepts electrons from complex 2 and transferred to complex 3. So common carrier between complex 1 and 3 and 2 and 3 is the ubiquinone. Location of ubiquinone within the membrane. Within the membrane, I think clear. And the location of cytochrome C is between complex 3 and complex 4. I will draw, see carefully here, all of you. Cytochrome C cytochrome C. That is the location. The location of cytochrome C is outer surface of inner membrane. Outer surface of inner membrane contains cytochrome C. Outer surface of inner membrane contain cytochrome C. So cytochrome C is present on the outer surface of inner membrane. Outer surface of inner membrane contains cytochrome C, a mobile carrier between complex 3 and complex 4. They take electrons from complex 3 and transfer to complex 4. This is the function of cytochrome C. What is the function of ubiquinone? Ubiquinone takes electrons from complex 1 and transport to complex 3. Similarly, it also accepts electrons from complex 2 and transport to complex 3. So these are moving carriers, mobile carriers. Now, clear all? Clear? Now let us see. I told you already, how many NADH are formed totally? 10 NADH. 2 in glycolysis, 2 in link reaction, 6 in NADH. Total number of NADH are 10 NADH. Now these NADH oxidized in the complex 1, first complex. Each NADH is oxidized in the complex 1, and at the end of the ETS, each NADH produces 3 ATP. Each NADH produces 3 ATP, sorry. And finally, how many FADH2 are produced? 2 FADH2 are produced, correct? And each FADH2 produces 2 ATP, correct? That is equal to 2 into 2, 4 ATP. Clear now? Here also 1 NADH 3 ATP, therefore 10 into 3, 30 ATP. 30 ATP. Therefore, total number of ATP is equal to 30 plus 4, 34 ATP. 34 ATP. Once again I will tell you, totally 10 NADH are formed. These NADH enters into ETS, starts with the first complex, and at the end of which we can say 1 NADH form 3 ATP. Like that, how many NADH are there? 10. 10 into 3, 30 ATP. Right. Similarly, 2 FADH2 are formed for each glucose molecule in Krebs cycle. Each FADH2 through ETS produces 2 ATP. Therefore, total number of FADH2 are 2. 2 into 2, 4 ATP. 30 ATP here, 4 ATP here, 30 plus 4, 34. Already 4 are produced directly. 34 plus 4, 38. Now we will see how they are produced. Now, first of all, I am taking NADH. 1 NADH, if I will take, then you can multiply, you will produce the 30 ATP. Okay. Each NADH oxidized here. See? NADH undergo oxidation it become NAD plus. So NADH oxidized in the first complex of ETS so that it becomes NAD plus. As a result, electrons are released. These electrons transported first FMN, then iron sulfur center, then from complex 1 they are accepted by ubiquinone, so that ubiquinone becomes UQH2, ubiquinol, it becomes UQ become UQH2. This UQ transfer its electrons to complex 3. In the complex 3, first they accept cytochrome B, then iron sulfur, then cytochrome C1, and then these electrons are taken by cytochrome C. Cytochrome C migrates from complex 3 to complex 4. Okay, these electrons are accepted by, in the complex 4, first copper A, then cytochrome A, then cytochrome A3, and finally copper B, and from here it is accepted by half O2 and also protons from the stroma plus electrons here and form water molecule. I hope you understand. The role of oxygen is in the fourth complex. The role of oxygen is at the fourth complex. Correct? So, the electrons from the complex 4 accepted by oxygen and form water. This water is called metabolic water. Along with the electrons, hydrogens, protons, electrons, protons and half O2 combinedly form water molecule. That means for each NADH oxidized in the complex 1 forms one water molecule, and the terminal hydrogen acceptor is oxygen. Terminal electron acceptor is oxygen. Terminal hydrogen acceptor or electron acceptor is oxygen. So the role of oxygen is present in which complex? The role of oxygen is present in the which complex? Complex 4. Okay? During this process, at 3 places here, 4 protons are transported. In the complex 3 also 4 protons are transported. In the complex 4, actually 4 has to be transported. Here 2 protons are used. Therefore, only 2 protons are formed. See carefully here. While transporting the electrons from one electron carrier to another electron carriers, at 3 places, protons are also transported across the membrane from matrix into intermembrane space. In the first complex, 4 protons are transported. In the third complex, 4 protons are transported. In the fourth complex, only 2 protons are transported because the remaining 2 are used in the formation of water. So finally, for each NADH oxidized, totally 10 protons are accumulated in the intermembrane space. 10 protons are accumulated in the intermembrane space. See all of you. Okay. NADH oxidized. Electrons are transported. First complex 1, then ubiquinone, then complex 3, then cytochrome C, then complex 4. Finally, the electrons from complex 4 are accepted by oxygen and form water molecule. Clear? During this transport of electrons, protons are also transported across the membrane from matrix into intermembrane space at 3 places. 4 protons in the complex 1, 4 protons in the complex 3, and only 2 protons in the complex 4. The remaining 2 here are utilized for formation of water molecule. Thus, at the end of this, totally 10 protons are accumulated for each NADH in the intermembrane space. Correct? Now, see what happens. More number of protons are accumulated in the intermembrane space. Correct? For every one NADH oxidized, 10 protons are produced. One more NADH, 10 protons. One more NADH, 10 protons. That means protons are accumulated across the membrane here in the intermembrane space. As a result, more protons in the intermembrane space and less protons in the matrix. As a result, proton gradient is established. Proton gradient is established. More protons, less protons. The difference of concentration of protons form proton gradient. Understood? Proton gradient. More protons, less protons. The difference create a proton gradient, and when this proton gradient breaks, ATP is synthesized. When this proton gradient breaks, ATP is synthesized. Okay? How the proton gradient breaks? See, more protons are there here. They want to move back into the matrix, but these complexes does not allow. These protons want to move back into the matrix, but only one-way transport. This movement of protons does not allowed by this complex except the complex 5. So these protons are allowed from intermembrane space to matrix. Therefore, these protons transport through the 5th complex into the matrix. First, these protons moves into the F0 called as transmembrane channel or proton channel, and from this F0 the protons are passing into the matrix through the F1. F1 is the head, F0 is the tail. So when protons are transporting across this 5th complex, first into F0 then into F1, in the F1 energy is liberated. Using that energy, ADP combines with inorganic phosphate and form ATP. So in the 5th complex, ATP is formed. Okay. In the 5th complex, ATP is formed and released into the matrix. I repeat once again carefully. More protons are accumulated in the intermembrane space. These protons moves into the matrix only through the fifth complex. First, the protons are passed into the F0 component. From F0 moves into the F1, and from F1 they moves into the matrix. While moving from F1 into the matrix, in the F1 energy is liberated. Using that energy, ADP coupled with inorganic phosphate and form the ATP. ADP coupled with the inorganic phosphate and form the ATP. This is known as oxidative phosphorylation. Totally, for each NADH, how many ATP are possible here? For each NADH, how many ATP are possible? 3 ATP. 3 ATP. One NADH can form 3 ATP. I told you already. One NADH, 3 ATP. Like that, how many NADH are oxidized for each glucose molecule? For each glucose molecule, how many are oxidized? How many are produced? Of course, 10. 10 into 3, 30. And one interesting point is, in this process, one complex is not involved. What is that complex? That is complex 2. In the complex 2, FADH2 is oxidized. In the complex 2, FADH2 is oxidized. FADH2 becomes FAD plus. FADH is oxidized in the complex 2 and releases electrons. These electrons moves into FAD, then iron sulfur complex. From iron sulfur complex to ubiquinone, so that ubiquinone becomes UQH2, ubiquinol. Now this transport to complex 3, first into cytochrome B, then iron sulfur, then cytochrome C1, then cytochrome C, then to the complex 4, and finally accepted by oxygen and water is formed. Same procedure. That means first complex is surpassed. That means in case of FADH2, first complex is not involved because it donates electrons to complex 2. From complex 2, electrons transfer to ubiquinone, ubiquinone to complex 3, then cytochrome C, then complex 4, and finally accepted by oxygen, and thus water is formed. So that means it missed the complex 1. Because of that, it loses 4 protons. So 4 protons. That means how many protons are possible for FADH2 in the intermembrane space? How many protons are accumulated in the intermembrane space from FADH2? Only 6. 4 plus 2. Because this gateway complex is not involved. Therefore, 4 protons are less. That means the energy efficiency of FADH2 is less. Only 4 plus 2, 6 protons are accumulated. From these 6 protons, the possible number of ATP are only 2. See, 1 FADH2 produces 2 ATP only. Like that, 2 FADH2 are formed. 2 into 2, 4 ATP. Which is more, as far as energy is concerned, which is more productive compound? NADH. 1 NADH, 3 ATP. 1 FADH2, 2 ATP. Why those less number of ATP? Because FADH2 oxidized, only 6 protons. NADH2 oxidized, 10 protons. Therefore, the efficiency of NADH is more as far as ATP is concerned than FADH2. So thus finally 34 ATP are formed through oxidative phosphorylation. That's our students today. This is about ETS. I hope you understand all. Thank you, and continue in the next class. Thank you. Bye.